Cytokines and the complement system. So cytokines are small soluble proteins that serve as chemical messengers of the immune system. If you remember learning about, like in your AMP class, the endocrine system, and how the endocrine system is a series of organs and they communicate with one another and with other parts of the body by producing hormones. The immune system has its own set of chemical messengers, and cells of the immune system, your white blood cells, are going to release these chemical messengers, and there's receptors for these chemical messengers in various parts of the body. Regulating the immune system has a lot to do with regulating where these chemical messengers are allowed to go, what they're allowed to bind to, and how much are released and when they're released. And if we have too much of the chemicals, too little of the chemicals, or not enough receptors that the chemicals are going to interact with and bind to, that's where we can get issues with our immune system. But in a normal, healthy individual, these cytokines are going to float around in the body, they're going to bind to their receptor, and then they're going to produce an effect in the cell. So whether it's going to cause the cells to make more copies of themselves, also known as like proliferation, or if it's going to cause the cells to deteriorate through apoptosis or programmed cell death. These chemicals have a wide range of activities in the body, and we'll go through some specific examples of what they can do. Cytokines can assist in both innate and adaptive immune responses. They are induced in response to stimuli. So sometimes cytokines are going to be released due to bacterial lipopolysaccharides. Sometimes flagellin can stimulate their release. Bacterial products, or the ligation of cell adhesion molecules. Cytokines can cause growth, differentiation, and gene expressions on many cell types, including leukocytes, or your white blood cells. The modes of action, so cytokines can go through autocrine, paracrine, or endocrine activity. Autocrine means that a cytokine is released by a cell and it actually acts on that cell. It acts on a self cell. Paracrine means that a cytokine is released by a cell and acts on a nearby cell. Endocrine would mean that a cytokine is released in one part of the body, travels through the bloodstream to another part of the body, where it finally has its effect. So here you can see a diagram of an antigen-presenting cell, APC, and you can see that this antigen-presenting cell has the ability to release a cytokine. In this example, it's IL-1 beta, and this IL-1 beta has an autocrine effect, so it's going to cause and stimulate that antigen-presenting cell further. This antigen-presenting cell that releases that IL-1 beta can also impact nearby cells. If it releases it into the bloodstream, it can have widespread effects in the brain or liver or other parts of the body. The receptors for these cytokines are distributed in various locations of the body. So a lot of people wonder, well, if cytokines are causing an issue, if cytokines are spiraling out of control, why can't we just block the cytokine? Well, if we block the cytokine and there's receptors for that cytokine all over the body, you just had a major impact on the entire body. So we really do have to evaluate, like, how many receptors are there and where are they located, and do we even know where all the receptors are today? The answer is no. We don't know as of today where all of these receptors are, but we have a good idea where many of them are, and we know that they have widespread activities around the body. So they don't just regulate the immune system, they also play a role in overall physiological function. Synergy means that two cytokines are going to work toward the same end goal. So they're going to work together to produce a common effect. Pleiotropy means that one cytokine has many actions, so it can do different things depending on the receptors in the cells that it binds to. Redundancy means that you have two cytokines that do the exact same thing. So they're not just working together toward a common end goal, they're actually doing literally the same thing. An antagonism is when cytokines work against each other. And we're going to get into that in more detail, but that is absolutely essential. There's cytokines that will induce the immune response, and there's cytokines that will dampen the immune response or decrease the immune response. And it's that delicate balance that we need in order to have a normal functioning immune system. A cytokine storm. So cytokine production leads to rapid generation of innate and adaptive immune responses. The regulation of cytokines can determine the clinical course of the infection. Hypercytokinemia, also known as a cytokine storm, can lead to shock, multi-organ failure, or even death. So it's a good thing that we have cytokines, especially the ones that are pro-inflammatory, because we initially want some inflammation. We want the inflammation to happen so that we get more cells to the area. We can clear up that infection pretty quickly if there is an infection, and we can start to repair the damaged area or damaged tissues or cells in that area. But we don't want that extended hyperactive state for too long. If we let cytokines build and build and build and create chaos and overstimulate the immune system in a particular area of the body, that can actually lead to excessive damage to our own cells. And that damage can then cause all of these bad things to happen, like shock, multi-organ failure, or death. So we need those anti-inflammatory cytokines to stop the pro-inflammatory cytokines. So this antagonism that I just mentioned, we need the ones to stop the other ones, and then our body can go back to normal. So initially, we want some inflammation, we want the cells of the immune system there, we want repair to happen, but eventually we need that to stop. And if it doesn't stop, that's when we can have prolonged issues. One specific example is if we talk about Lyme disease. So Lyme disease caused by Borrelia burgdorferi, which is transmitted by the tick. And it's a specific type of tick, the deer tick, and it's in specific regions in the world. So particularly Wisconsin is known for Lyme disease, and then over on the East Coast there's certain regions that have Lyme disease. But that type of tick transmits that type of bacteria. Now when that bacteria gets in your body, usually a tick has to bite you and be attached to you for more than 48 hours in order for adequate saliva and bacteria exchange to happen for you to even get infected. But the bacterium then goes into the body and it migrates to different tissues in the body, particularly the joints. And when it gets to the joints, you're going to have some local inflammation. You may have like flu-like symptoms, you may spike a fever. And pretty much at that point, the bacteria is killed, and the bacteria isn't found in the joints anymore. But because the bacteria was in the joints, certain components of the bacteria stay behind, and we can get a prolonged inflammatory state. So the cytokines in the area keep being stimulated. They keep being released and stimulating the local cells and cause prolonged inflammation. And that inflammation can go away and then come back later and go away and come back later, even though the bacteria is dead and gone and it's not there anymore. And sometimes we can't even find the genetic material for the bacteria anymore. So having that prolonged pro-inflammatory state can lead to a lot of cell damage and destruction. And that's basically how Lyme disease causes joint issues for a person's lifetime after they get cleared of the infection. It's not that the bacteria is there, it's that the bacteria was there and left something behind that really messed up and interfered with the immune system's normal function. So there's different categories of cytokines: tumor necrosis factors, TNFs, interferons, IFNs, chemokines, transforming growth factors, or TGFs, and colony stimulating factors, CSFs. It is important that you know the abbreviation. You will be asked questions, what is a TNF? And you'll have to know what that is. A lot of times on like medical reports and stuff and patient results, you're just going to see the abbreviations and you need to know what they mean. So here's a list of some of the major pro-inflammatory cytokines and anti-inflammatory cytokines. So pro-inflammatory are going to induce immune responses. Anti-inflammatory are going to decrease or depress immune responses. And it's a delicate balance that we need to maintain a normal functioning immune system. So TNF-alpha, IL-1, 6, interferon gamma, those are our pro-inflammatory cytokines. Anti-inflammatory cytokines would be TGF-beta, IL-10, IL-13, and IL-35. IL-1. IL-1 is a pro-inflammatory cytokine that's produced by antigen-presenting cells early on in an infection. It acts as an endogenous pyrogen. Endo means from within or from inside. Endogenous means it was generated from within, so it comes from that person. Pyrogen, pyro is referring to fire, and gen is like the generation of something or making something. So fire is heat, hot. Think about a fever. So endogenous pyrogen is a molecule that's from within the body that induces the formation of fever. It induces fever in the acute phase response through its actions on the hypothalamus. It induces the production of vascular cell adhesion molecules, chemokines IL-6, which includes IL-1 alpha, beta, and IL-1RA. It's an antagonist. It blocks the IL-1 receptor. So all of those are categorized under IL-1. IL being short for interleukin. Tumor necrosis factor alpha, TNF-alpha. It is a pro-inflammatory cytokine. It's the most prominent of the TNF family. It exists in membrane-bound and soluble forms, causes vasodilation and increased vasopermeability. Vasodilation means it causes the blood vessels to dilate or to get wider. And when you cause those blood vessels to get wider, they inadvertently also become leaky. So vasopermeability is talking about the penetration of solutions out of the bloodstream. Vaso, referring to the blood vessels. Permeability is the ability for something to move through a membrane. It's triggered by the presence of lipopolysaccharide, which is found in gram-negative bacteria. It activates T cells, but can lead to an overreaction, such as what's seen in septic shock. IL-6 is a single protein produced by lymphoid and non-lymphoid cells. It's part of the cytokine cascade released in response to lipopolysaccharide. It's pleiotropic and plays a role in acute phase reactions. It's triggered by IL-1, and elevated levels may follow psychological stress, leading to an increased risk of atherosclerosis. Atherosclerosis is talking about thickening of the arterial walls or plaque formation, which leads to a constriction of blood flow in the area. So plaque forming inside of the blood vessels. Chemokines. So chemokines are a type of cytokine. So cytokines referring to all of the chemicals. Chemokines are a very specific type of cytokine. And what chemokines do is they promote the migration of different types of blood cells or white blood cells toward the source of where it was produced. In other words, they promote chemotaxis. Chemo is chemical, taxis is movement. In other words, movement in response to a chemical. So a cell is going to release a cytokine or a chemokine, and white blood cells are going to kind of follow the trail. If you've ever seen ants and you wonder how ants stay in a perfect line, and then when one of them finds food, they kind of lay down a chemical trail, and then they all follow that chemical trail to the food and back home. It's kind of the same thing. So a cell will find an area of inflammation or damage and realizes that, hey, we need backup here to help clear up this damage. So it's going to start releasing chemokines, and those chemokines are going to be like chemical messengers, and as soon as certain white blood cells pick up on that scent of those chemokines or get activated by a chemokine binding to its receptor, it's going to follow that chemical back to its source, and then it's going to be migrated toward the source of the damage. They're classified into 4 families based on the position of the N-terminal cysteine residues. So there's CXC, CC, C, and CX3C. They're involved in the initiation and development of the inflammatory response in numerous disease processes. Combined with cell adhesion molecules, facilitate the extravasation of leukocytes into the tissues in areas of inflammation. TNF-alpha and IL-6 induce chemokine production. Transforming growth factor beta, or TGF-beta. It's composed of 3 isoforms, B1, 2, and 3. It induces anti-proliferative activity in many cell types. It regulates cell growth, differentiation, apoptosis migration, and the inflammatory response. And it helps to downregulate the inflammatory response when it's no longer needed. It inhibits the activation of macrophages and the growth of different somatic cell types. It functions as an anti-inflammatory factor for mature T cells. In activated B cells, it inhibits proliferation, so it stops them from making more copies of themselves and spreading. And it may function as an autocrine regulator to limit the expansion of activated cells. Type 1 interferons, IFNs, IFN alpha and IFN beta, they interfere with viral replication. They're produced by dendritic cells and induce production of proteins and pathways that interfere with viral replication and cell division. They activate natural killer cells, because if you remember what natural killer cells do, they are going to destroy any cell that looks weird, particularly cancer cells and virally infected cells. They enhance the expression of MHC class I proteins. Well, why MHC class I? If you remember from the previous lecture, your normal self cells that get infected with a virus, they are going to chop up that virus and they're going to hold up a chunk of that virus to your immune system. Now, the arm that they hold that up to the immune system with is MHC class I. That is then going to trigger cytotoxic T cells to bind to it, so that when the cytotoxic T cell sees the MHC class I, it knows that it can kill your infected self cell. They're active against certain malignancies and other inflammatory processes. Cytokines and adaptive immune response. They're mainly secreted by T cells, especially helper T cells, or Th cells. They affect T and B cell function more directly than cytokines involved in the innate immune response. There's 3 main subclasses of CD4 positive T cells: Th1, Th2, and Tregs, or T regulatory cells. Th1 cells promote cell-mediated immunity, especially in response to intracellular pathogens like viruses and bacteria. So if you remember from previous lectures, I talked about how helper T cells help activate other cells of the immune system. So a Th1 specifically would help activate like our cytotoxic T cells that are going to then start to attack virally infected cells. Key cytokines include interferon gamma, IL-2, and tumor necrosis factor alpha, or TNF-alpha. Primary role, activate macrophages to enhance their ability to phagocytose and destroy pathogens. Promote the production and activity of cytotoxic T cells, in other words, those CD8 positive T cells, to kill infected self cells. Help in the defense against intracellular pathogens. Mycobacterium tuberculosis, viruses. Chlamydia is another intracellular pathogen. Th2 cells support humoral immunity, are involved in the defense against extracellular pathogens such as helminths. These guys are going to stimulate B cells to produce antibodies, particularly IgE, which is important in the allergic response and defense against parasites. They promote the recruitment and activation of eosinophils and mast cells, also play a major role in mediating allergies and parasitic infections, and drive immune responses involved in allergic reactions. For example, asthma and hay fever. Key cytokines include IL-4, 5, 6, 10, and 13. Regulatory T cells, or Tregs, maintain immune homeostasis and prevent autoimmune responses by suppressing excessive or inappropriate immune reactions. Key cytokines are IL-10, TGF-beta. Primary role, suppress the activation and proliferation of other T cells, both Th1 and 2, maintaining tolerance to self-antigens and preventing autoimmunity. They inhibit inflammatory responses and promote immune regulation to avoid tissue damage during infections. This is the key part. We want that initial inflammation, that initial immune response to clear the pathogen, to fix the problem. But then we want it to stop. If it doesn't stop, then we will continue to have excessive activation and tissue damage and problems and inflammation, and we don't want that to happen. These help prevent that from happening. It helps regulate the balance between immune activation and tolerance. So the Th1 cytokines, interferon gamma, stimulates antigen presentation by class 1 and 2 MHC molecules, activates macrophages to stimulate phagocytic and cytotoxic abilities, regulates the action of CD4 positive helper T cells and CD8 positive cytotoxic T cells or lymphocytes, and natural killer cells. IL-2 is known as the T cell growth factor. It drives the growth and differentiation of T and B cells, induces lytic activity in natural killer cells, and can activate proliferation of Th2 cells. Lytic means to break apart or cut up. So lytic activity is talking about their ability to cut things apart and break things up. Th2 cytokines are IL-4 and 10. IL-4 helps drive antibody responses in various diseases, allergies, autoimmune diseases, parasitic infections. And the receptor is expressed on lymphocytes and various non-hematopoietic cell types. IL-10 has anti-inflammatory and suppressive effects on Th1 cells. It's produced by monocytes, macrophages, CD8 positive T cells, and Th2 CD4 positive T cells. It inhibits antigen presentation by macrophages and dendritic cells. It serves as an antagonist to IFN gamma to downregulate the immune response. So the cytokines for Tregs are CD4, CD25, and T cells. Helps establish peripheral tolerance to various self-antigens, allergens, tumor antigens, transplant antigens, and infectious antigens. Produce TGF-beta that suppresses other T cells. Induces IL-10, TGF-beta expression in adaptive TR1 cells in the peripheral circulation. Immune suppression. So T cell suppression occurs through IL-10 inhibition of pro-inflammatory cytokines, inhibition of co-stimulatory molecule expression on antigen-presenting cells. TGF-beta downregulates antigen-presenting cell function and blocks proliferation and cytokine production by CD4 positive T cells. The immune response is downregulated and chronic inflammation is prevented. Th17 cytokines have a role in host defenses against bacterial and fungal infections at mucosal surfaces. It involves Th17 cells secreting IL-17, it's a family of cytokines to promote continuous recruitment of neutrophils. Promotes the release of antimicrobial peptides, and when dysregulated, have been implicated in pathogenesis of multiple inflammatory diseases and severe autoimmune conditions. So what I would do with all of this information, because it is a lot of information, I would draw a picture. So in the previous lectures that I gave, I had pictures and diagrams, and I showed how a B cell is activated and what it turns into once it's activated. Then I showed how a helper T cell and a cytotoxic T cell is activated and what happens after that. Well, now with that very same diagram, you can add in these cytokines, and you can say cytokines are gonna, and you can have like an up arrow for they increase the activity of these cells or they decrease the activity of these cells. And you can take those helper T cells a step further, and now you can talk about Th1 cells, Th2 cells, and then those Tregs, and you can draw those out on a picture. I am a really visual learner and I like seeing things and how things all put together. I'm not very good at just memorizing terms or definitions that come my way. So if I have it all diagrammed out, then I can kind of make sense of it. And even if I don't remember everything, I'm really going to remember where I had it on my diagram, and that can help bring back memories when I'm taking a test on this stuff. So just a bit of advice for how to even start to study for this. Clinical detection of cytokines. So ELISA spot assay employs the ELISA technique on in vitro activated peripheral white blood cells. There's also multiplexed ELISAs that can detect 12 to 25 pro and anti-inflammatory cytokines in one reaction. Microbead assays allow for the simultaneous detection of multiple cytokines in a single tube. All right, switching gears a little bit to the complement system. So complement is a series of more than 30 soluble and cell-bound proteins that interact to enhance host defense mechanisms against foreign cells. So complement is a little bit different than cytokines, because cytokines are like the chemical messengers of the immune system. Complement is like the little bullets of the immune system. So if we had if all of our white blood cells were carrying guns and shooting off little bullets, that would be like what complement is. Complement is going to be able to coat pathogens and target them to be eaten by your immune system. They can also form these things called membrane attack complexes, where you start with one complement protein and then a complement, they call it a complement cascade. So this complement protein gets broken apart into these two pieces, and then those pieces get broken apart and get broken. Eventually, you end up with these pieces that are able to embed themselves into bacterial cell membranes, and they can poke holes in the bacterial cell membranes, and that causes the bacterial cells to die. So the complement is different from cytokines, because cytokines are going to be our chemical messengers. They're either going to stimulate other cells to be activated, or they're going to tell those cells to calm down and stop being so hyper and active. So they're going to mediate those responses. Complement is really like the weapons of the immune system. They're little chemicals, little proteins that are going to do things to primarily bacterial cells, and then other types of functions that we'll get into in just a little bit. So most plasma complement proteins are synthesized in the liver, with the exception of C1 components that are produced by intestinal epithelial cells, and factor D that's made in adipose tissue. So what complement proteins can do? They can act as opsonins. Opsonization means coating. So in a previous lecture, I talked about the function of antibodies and how antibodies can coat a pathogen in order to clear it from the body. So complement actually sticks to the pathogen first, and then the antibody will stick to that complement protein, and then it makes this huge big clump, and then agglutination happens, and it can be cleared from the body a little bit faster. But opsonins, that's just meaning that they're going to coat the surface of a cell. Now, bacteria need to be able to stick to you to establish themselves in you. If you have free-floating bacteria, they can still cause problems. But freely floating bacteria can easily be filtered out of you through the spleen if they're in the bloodstream, or in the lymphatic system, they can be filtered out in a lymph node. But if the bacteria are able to stick to your surfaces, then they can establish themselves, they can start to replicate, and they can start to produce all the harmful things that they produce that are going to digest your tissues. Because the goal of the bacteria is really to get your nutrients so that it can survive. And in the process of doing so, it's going to kill your own cells so that it can eat those nutrients. So if we have these proteins that are going to coat the surface of the bacteria, it can't stick to you. And it's just going to be flushed through your bloodstream or flushed through your tissues and your lymphatic system. That's the system that's going to be absorbing all of that tissue fluid, cleaning it, and returning it to circulation. So if the bacteria can't stick to you, it's not going to be able to really cause significant infection. Specific receptors are present on phagocytic cells. Metabolism and clearance of immune complexes are enhanced, increase vascular permeability, recruit monocytes and neutrophils to the area of antigen concentration, and triggers secretion of immunoregulatory molecules that amplify the immune response. There are 3 pathways for complement activation: the classical, lectin, and alternative pathway. In the classical pathway, it's triggered by IgG or IgM binding to the surface of pathogens. In the lectin pathway, it's activated by carbohydrates that are present in microbial cell walls. In the alternative pathway, it's triggered by bacterial and fungal cell walls, yeast, viruses, tumor cells, and certain parasites. Multiple pathways can be involved, but the end product is lysis of the invading cell. Lysis again means to break apart or cut up. I talked about that membrane attack complex, how these complement proteins get broken down and exchanged into different pieces, and eventually in the end, they're going to combine into this membrane attack complex and literally puncture a hole in the cell membrane or in the cell wall of an invading pathogen. When you puncture a hole into a cell, it's going to rupture. It's going to cause lysis of that cell. There is a separate video that I've included that shows complement activation. I find that sometimes animations explain things a little bit better than what you would see in a 2-dimensional picture. So I could bore you and go through all of the steps of complement activation, or you can watch just a really cool animation about it and get a better picture. All right, so hopefully you take some time to watch that video that goes through how complement is activated through the classical, lectin, and alternative pathways. Laboratory detection of complement proteins. So techniques involve the measurement of components as antigens in serum, the measurement of functional activity, immunologic assays of individual components, radial immunodiffusion, or nephelometry. All of those techniques should be familiar from clinical chemistry. We can also measure the classical pathway. So we can measure the amount of lysis that happens. There's a hemolytic titration CH50 assay. CH50 tests for the lysis of liposomes, radial hemolysis in agarose plates. And then there's alternative pathway assays, AH50, ELISA, and we can test systems addressing all 3 pathways. And feel free to read through the summary. That sums up the lecture for today.